in the plane. Well, it is really a pleasure to be here with the colleagues around the world. And this is not the first time I'm in this series of the meeting, but this year is particularly important and with in person. So as you mentioned, I'm a plant pathologist. So you will see lots of the terms in plant pathology here. This is my email address. Any question, particularly comment, will be very much appreciated regarding the presentation. Well, if you wonder where I am, here it is, Illinois. If you don't know Illinois, know Chicago. Chicago is right here, is in Chicago near the lake. And most people in the world know Chicago better than Illinois. But Illinois is part of Chicago, and we are right here. University of Illinois, Urbana-Champaign, two twin cities side by side. For example, I work in Urbana, live in Champaign, you know, just one side of the street, Urbana, another side is Champaign. It is the biggest university of the state of Illinois. So everybody knows Apple, and we eat apple maybe every day, maybe, you know, every some days. And we want to eat healthy apple. This is what considered healthy apple. In fact, this is kind of new variety. I'm not going to mention variety rather than I'm going to mention some kind of new kind of apple, but that's what it is. Anybody had Honeycrisp? This is Honeycrisp. Honeycrisp apple, you have? Anyone else? Oh, you had Honeycrisp is so sweet. It's so sweet, attracts all sorts of insects and everything, including pathogens. And it is kind of new variety or new cultivar, if you wish. And it was developed at the University of Minnesota, far northern here. It was developed here. It's cold area. So then you would guess what kind of problem we're going to have with this particular cultivar variety. How many of you are plant pathologists? Oh, you are plant pathologist? Oh, good plant pathologist. How many of you have experience on plant and apple diseases? Oh, great, oh, my goodness. Okay, just to mention, you know, if you go compendium of apple diseases, you will see lots of diseases up there, but these are the major ones. Fire blight. Fire blight is a bacterial disease in everywhere. And then we do have scab. It's a fungal disease also everywhere. And we do have rust. Rust are not everywhere, but most places. Then we do have sooty blot and fly speck, two different diseases, but they occur together, mainly on the fruit. Then finally, we have fruit rots, 3 fruit rots. These are very important. Anybody knows where is the origin of apple? Where the apple came from? Not from Singapore, not from Indiana, not from India. Where is the origin of apple? Go ahead.
I said Garden of Eden.
Where?
Garden of Eden.
Origin of apple. Origin of apple is Kazakhstan. Origin of apple is Kazakhstan, Central Asia. You know, at the time of Soviet Union, they built a big town called Alma-Ati. Alma-Ati, Alma means apple, Ati means father, fatherland of apple. Any other place in the world you see, somewhat came apple from Kazakhstan, then evolved. So I have been in the mountains where the really original apples were. And right now, if you go Kazakhstan, one of the poorest country in apple production, unfortunately, was supposed to be a leader in apple production, breeding, pathology, entomology, horticulture, and so on. I believe there's going to be somebody from Kazakhstan in the meeting. Is there anybody? Anybody from Central Asia? No. Okay, if you know where Kazakhstan, and anybody from China? Anybody from China? Who is from China? If you consider China, if you go northwestern province, Xinjiang, northwestern province, Xinjiang, capital is Urumqi. Across the border is Kazakhstan, not too far from the mountains of origin of apples. Is fascinating. If you get a chance to travel, you will see a lot. I have been there 5 times. I mean, not particularly for apple, but for different reasons. Here it is the pictures of fire blight. Pathologists know. This is the fire blight, the shoot blight. Fire blight have different symptoms. This is the shoot blight. These are from Illinois. You see, black, black, black. If you want to see if this is fire blight, you have to go one of the shoots and see shepherd hook. A tip of the infected shoots bends. That is indicating a fire blight in the field. I do have lots of pictures. This is the canker, another major symptom of fire blight bacterium. In fact, this is from Kazakhstan, from Almaty area. Supposed not to be there. What happened? They brought some cultivars from Europe and they brought pathogen with that to Kazakhstan. Who knows what's going to happen for the future? You know, this gene pools in Kazakhstan, variety of the genes. Anybody involved in breeding a program in apple goes Kazakhstan, but right now it's a little bit complicated. So this is the scab. Also, I believe you are from Australia, right? If you go Australia, there's a scab. If you go Brazil, there is a scab. If you come to, for example, Illinois, there's a scab. Europe, very similar, everybody. Major fungal diseases of apple. This is the rust. This is the rust called the cedar apple rust because it has 2 hosts. On cedar, they move from the cedar to apple. Wherever you have cedar, potentially you're going to have this disease too. Only single cycle of this disease is there. But you can see the magnitude of the important. Sooty blots, fly speck. These are sooty blots, all of them. This is fly speck, looks like a flood eye, you know, the flies. All together, almost more than 99%, they are together. They are superficial. They are not terribly difficult to control this disease, but is there. All superficial. If you remove the skin, there is nothing in their flesh. It's just superficial. These are fruit rot, 3 major fruit rot. We have more than that. We have, for example, if you come to United States, major apple growing state is state of Washington, not the state of capital. In Washington, D.C., state capital, they just grow politics, not apple. But if you go state of Washington, they grow apple, lots of apple, probably biggest apple growing center in the world, as far as I know. I know Chinese has probably 3-4 million apple orchard, but that's okay. This is the one giving us problem. Not us, but in the United States. In Korea, for example, is a big problem, and several other places. It is so important. I just prepare the fact sheets. You can see 1 lesion, 2 lesion, several sites of infection, and infected apples on the ground. 100% of the losses of the fruits is not uncommon. You can go, and particularly with the Honey Crisp, the new variety, you see apples are on the ground, nothing to harvest. This is the sad. I do have 3 responsibilities: teaching, research, and extension. Our extension is not extension like in India or like in Central Asia. All of us at the university, land-grant university, have at least 2, rather 3 responsibilities. For example, if you go India, extension is different organization under Ministry of Agriculture. And in the United States, we do take care of the research, then extension. We identify the problem, try to solve the problem, and take the message to end users. That's what I have to do. Then that was my job, to go and develop this one. The most important step in controlling disease, I'm going to say managing the disease. I don't believe the control of the disease. Managing the disease is accurate identification of the causal agent. If we don't know what is the causal agent, we're going to be treating wrongly. Looks like taking a pain reliever rather than really taking a real medicine aiming the pathogen. That's what it is. We did here. During 2000, I work on apple disease and the other crops too. In 2000, we did not have Honeycrisp variety or Honeycrisp cultivar. Then we investigated diseases and the causal agent, and particularly epidemics or epidemiology of, let's say, fire blight, scab, and the others, and pretty good, then came up with what? Came up with a very good, realized that this disease, fruit rot, is basically related to the moisture on the apple. If there is no moisture on the apple fruit, there's going to be no rotting. Then that was great. Then we developed a model using this data logger in the canopy, worked very well. What was this based on? Was based on spray the trees until about 10 days after petal fall. Whatever you do during bloom, after petal fall, this is called the first cover spray. After that, we have to accumulate 175 leaf wetness. If anyone really curious what it is, I can explain later on. And we were able to reduce almost 50% of the spray. In Illinois, the growers spray about 8 times between, let's say, May to September. And we were able to cut it down to about 4 to 5. Very significant accomplishment. Then suddenly popped up this guy, Honeycrisp. Honeycrisp is like honey and crispy. Then came up, then the beetle rot. Nothing, no fungicide, nothing could stop it. And we did not know even how it is working, how it is developing. We started really exploring or searching about the disease. This is a map of Illinois. We started 2019. In fact, we started 2017, did not work. Then mainly data gathering was from 2019. You see, this is Illinois, and these are the apple orchard. We surveyed 24. Out of 24, 14 had bitter, bad bitter. Where they were in the southern Illinois, not in northern Illinois. Northern Illinois is very close to Minnesota, where Honeycrisp was developed, released. Then this was the first year. Second year, 2020, pretty much similar, but we realized pathogens are moving up too, very slowly. You see, first year we had almost nothing. In 2021, out of 30 orchards, 67% had beetle. And then we continued on 2021, very similar. So pretty much now we knew that how bad it is, what is the incidence, what is the severity of the disease, and where the disease are mainly developing in southern Illinois, western Illinois. This is the major apple growing area, and this is next to Chicago. All of the apples are for fresh market. Then we collected 270 isolates of the pathogen. Then we saw different color. What does it indicate? They are genetic variation among the isolate. We have to be very, very careful. Then you see this one, these are from the top of the Petri plates, and these are from the bottom of the Petri plates. All we could say, these are not the same. They are genetic variation among the isolate. Then we continued on. Originally, what we knew and what other people had reported in the compendium of apple diseases, they were Calatatricum acutatum complex. They were not saying the Calatatricum acutatum because there were lots of variation. They said complex. Then another group, Calatatricum pilosporidius complex. This complex is okay, but was difficult for us to focus on developing effective management. What we did, we used primers. These are the primers we use. In PCR with 25 microliter of complex. Then what we ended up, 3 different species in Illinois, almost brand new. They are not all from us, but at least in the, we say Midwest of Illinois. We were the first one to really recognize this. This was Calatatricum fiorinum. This was really Calatatricum acutatum species complex. But this was the accurate species name. Then Calatatricum siamens in the Clostridium species complex. Then this is brand new, first time in Midwest of US, not in the Northeast. Northeast had been identified this one about 3-4 years before we did in the Midwest. 3 different species. Here we go. Right now, we know the species, we know the severity of the disease, we know the incidences, we know also the susceptible varieties. Then we started from here. Here we go. Species, where are they? Look at this one. Calatatricum siamens. Calatatricum siamens. Calatatricum siamens, also Calatatricum here is, well, these are the species. In some of the apple orchard, we had all 3 species. Some of the apples, single apple fruits, we had 2 and very rarely 3 species. So we did not know that. I mean, not a single species, so complex. Anyway, you can see here, if you add them together this is from 2021 21 orchard, but if you add them together, 18, 11, 29, 30, meaning that in each apple or in each orchard, more than one species. Then everything was clear. Then we had to come up with a solution with delivering messages to the user how to take care of this disease. That was the hard part. And we had to continue on. These are the, you see the three, on Calatatum, Acutatum species, and you see in the Midwest, we pretty much plugged in, and you can see very similar for Clostridium. You can see how much genetic variation among the isolates. All started with a new apple variety, new apple cultivars, Honeycrisp. So we had to start life all over. You can see we inoculated the apple fruit in the lab. This is Calatatum fiorina, Calatatum chrysophyllum, the first time in the Midwest, Calatatum siamens. Then we inoculated three cultivars, three varieties of the apple are susceptible. Was Honeycrisp, Empire, and the Golden Delicious. You can see, for example, in the pathogenicity, for example, here, let's say Fiorina. This is Fiorina. See, Fiorina is sporulating. Its lesion is a little different from variety to variety, but it's sporulating. Chrysophyllum is growing very slowly. And Siamens is growing very rapidly and depressed area. Then we collected on the pathogenicity too, and very good. Then we have to come up with effective fungicide. Without the fungicide, you cannot grow apples. You cannot harvest apple anywhere in the world. You have to use the fungicide. You may say, oh, there are some resistant cultivars. Yes, they are resistant cultivars, but the growers do not grow them. Growers really produce the variety they can easily market. Market demands what? Market demands Honeycrisp. They're not going to grow Red Delicious because value is less than 50%, maybe 30%. They're going to grow something they can get more money, more value. So we started checking fungicide. More than this, we did more than this. But if you want to see finally what fungicide we came up in the lab for testing in the field, in the orchard, these are the ones. This is the common name, and these are the commercial names. You see, for example, Capton is Capton. But this is, where is this one? Miravan. This is relatively new compound, new fungicide, less than 10 years old. You see, this is a combination of the two, fluxapyrrole plus pyraclostrobin. So in the lab, we pretty much came up with what is working. And the first year in the orchard, we came up with the selection of the fungicide for susceptible varieties. Here, I'm going to present only 2 years in the field, but this is from the lab. You see, in the lab, we call EC50. The fungicide reduce colony of the fungus by 50% after certain days. We call EC50. We did. You can see Aprovia. Aprovia, this is the commercial. If you want the common name, you go back to previous slide. This was good. And you can see Chrysophyllum needs a little bit higher. And you can see tree isolates from Illinois, from the central, from the north, and from the south, and you can see what is going on. For example, one of the compounds we were hoping that to have a good effect was prophyte. Prophyte is potassium phosphate. You see, was not very good. But in comparison, look at the marijuana. It's really very little, very effective. This was very good and very encouraging in the lab study. Then we had to go to the orchard. Only orchard we had available for the university was Honeycrisp. Honeycrisp, susceptible cultivars. We did. All trees were sprayed with Fantalis plus Mancozeb, alternated with Inspire Super plus Mancozeb until bloom. When bloom started, we had to go different. From bloom to harvest, trees were sprayed with different fungicide. That was the testing. What were they? Here they are. Treatment rate, percent fruit with the bitter rot in 2 columns. You can see up here. Control, the first year, 2019, 51% infected. Phosphite. Phosphite by itself, not. Prophyte and Reliant is kind of potassium phosphate. By itself is not effective, but the rest of them perfect. We said, this is good. Let's go to the second year, 2020. We did a combination. I'm not going to spend time go to a combination. That's the file is going to be available for everybody. If you just send me an email, I will be more than happy to send you PDF file, even more information. Then we started spraying from July 21st, went out, control plots, 100% losses. The rest of them, perfect. This is 10 days spray, 14 days, 14 days, all 14 days. That's what growers needed. Then that was not enough. Just 2 years. We went to 3 years, 2021. In 2021, we found, yes, this is the last spray was August 17, but in September 5, we detected disease in some of the plots. If you calculate, this is more than 14 days. What we found, these combinations are perfect, but only they can protect fruits for 14 days, not 16 days, not 17 days. If you planning, if you are grower, growers know what I'm talking about. If little infection, they just take to the cold room and then to the market, then there will be more disease in the fruits. Only 14 days. These are perfect, only 14 days. If this is going to be harvesting, after 15, 16, 20 days, you have to spray again. And then, what are these? What are the important in pathology? We do have, these are the common name, these are the commercial name, and these are the frac code. Is there anybody does not know frac code? Fungicide Resistance Action Committee. Fungicide Resistance Action Committee. This is what is missing in the developing countries. When I go, for example, Kazakhstan or even Eastern Europe, they don't have much information about this kind of stuff. Then they use fungicide after fungicide, they get resistance in the pathogen. And another thing is PHI, days, number of the days to harvest, PHI, pre-harvest interval. You see, for example, Aprovia is great, but you can't spray within 30 days to harvest. So with this one, with all this finding, we found most susceptible cultivars: Empire, Honeycrisp, and Golden Delicious. Moderately susceptible cultivars, so Jonathan, Gala, and Fuji. Less susceptible, Macintosh, Granny Smith, and others. Everything. This was really good finding for Illinois apple growers. I would say beyond Illinois apple growers in Wisconsin, apple growers in Indiana, in Iowa, and so on. And this is my recommendation to the growers. Try to do cultural practices as much as possible. What is that? Remove the mummies, the dried apple from one season to another season. They carry fungus. And then the burn whatever dead woods are. Dead woods just carry the fungus from one season to another season. Began spraying at pedal fall for this bitter rot disease. And then go here. Marijuana, this is the right now in Illinois, they're using Marijuana plus Captain, alternated with Profite plus Captain. Works perfect. Or Marijuana plus Captain, alternated with Topsonin plus Captain, or Zyron plus Captain. Begin spray after pedal fall, spray at 14 days interval. If happened, we have rain after rain, fungicide are washed off, you may need more than 8 spray, then use Aprovia plus Zyron in early season. As you saw, Aprovia has 30 days PHI. You can't use Aprovia in late August or September because that's harvesting season, but you can use in June or early July. With this one, thank you for your patience, and I would appreciate if there is any comment to really improving this recommendation or any question might be, I will try to answer my best.